As we begin working with microorganisms in the laboratory, we need to know appropriate inoculation and isolation techniques. Microorganisms need nutrients, and they either get these nutrients from the environment if they are free living, or from their host organism if they live at the expense of another organism. When we try to grow them in the lab, we have to provide the nutrients using artificial media. Media can be classified based on its physical type. Solid media is called agar because agar-agar is the solidifying agent. This is an extract of seaweed. It solidifies at 42 degrees centigrade and melts at 100 degrees centigrade, so it stays solid at incubator temperatures. A broth is simply a liquid media. Semi-solid agar has half the agar content of regular agar, and we use it primarily when we're looking at motility. Media can also be classified based on its function. An enriched media has extra ingredients for fastidious organisms. These are organisms that require extra nutrients. Selective media has ingredients that inhibit the growth of an organism or a group of organisms. This allows other organisms to grow more efficiently. Differential media allows for separating organisms into groups based on some special ingredient in the media. When we detect microbial growth on agar, we look for a mass of cells. This is called a colony. If the colony is sitting all by itself, it's said to be an isolated colony and considered a pure culture. In broth, we look for the presence of cloudiness or turbidity. Inoculation of media is simply placing bacteria on media using sterile inoculating loops or needles. Aseptic technique is used when handling plates and tubes so that we don't introduce contaminants into the culture. Isolation techniques are special inoculation techniques that allow us to separate cells. If we have an individual cell that multiplies, it will produce a visible mass of cells. This is known as a colony. An isolated colony then would represent the reproduction of only one cell and would be a pure culture. There are different types of isolation techniques. The streak plate is when organisms are spread on the surface of the plate using the streak dilution technique. This is a technique that's a series of streaks designed to separate bacteria into individual cells. The pour plate is when organisms are introduced into liquefied agar. The organisms will mix in the agar and be completely separated. And the spread plate, which is one we will not do in laboratory, is when a specific volume of material is placed on the plate surface and then spread around. Just to review some aseptic technique, you're going to hold your loop in your dominant hand like it's a pencil. You'll sterilize the loop in the bacticinerator because it will glow 3 seconds and you will watch it glow for 3 seconds. When taking lids off of tubes, you will use your pinky finger to grasp the lids on the tubes and keep the lids in your hand until you are finished with that tube. The lids cannot be put on the table. When working with agar plates, you keep the agar side up. And you also keep the agar surface of the plate as perpendicular to the table or surface as possible. To inoculate a broth culture, you're going to use a sterile loop to pick up the bacteria you want to culture. You will need to remove the lid from the broth tube using your pinky finger and keeping the lid in your hand. Place the loop into the broth and swish it around a little bit. You will then put the lid back on the tube, put the tube in a test tube rack, and incubate the tube at 35 degrees centigrade for 18 to 24 hours. To inoculate an agar slant, you will once again pick up some bacteria with a sterile loop. You will remove the lid from the tube using your pinky finger. You will insert the loop to the base of the slant and simply drag the loop up the surface of the agar. Just let it glide up the surface of the agar. You don't want to puncture the agar or dig into the agar in any way. To inoculate a streak plate, you will use your sterile loop to pick up the bacteria to be cultured, and then you will lift the lid of the plate and streak half of the agar surface with the loop. You want to use back and forth movements that are fairly close together. You will return the agar plate to the lid and sterilize the loop in the bacticinerator. You will then lift the agar plate and using the sterile loop, streak one time into your previous streaking. You will then streak about a quarter of the plate, again using close together back and forth movements. Return the agar plate to the lid and sterilize the loop in the back decinerator. Once again, lift the agar plate and using the sterile loop, streak one time into your second streaking. Then streak the remaining 1/4 of the plate using again close back and forth movements. Return the agar to the lid and sterilize your loop. Label the plate, and you always label the agar containing portion of the Petri dish. You will then incubate the plate upside down in the incubator for 18 to 24 hours at 35 degrees centigrade. When you're inoculating a streak plate, it is very easy for the wire loop to cut or gouge the agar. This is considered bad technique, so to prevent doing that, you want to hold the loop parallel to the agar. That is, the loop should be parallel to the agar going back and forth and not up on its side. If you use this, it's like using a cheese slicer on the agar. The loop will sink into the agar and you will cut divots and digs out of it. Tubes of agar have been melted by heating them to 100 degrees centigrade. They have been kept liquid by placing them in a 60-degree water bath. By cooling them down to 60 degrees, we will not kill organisms when we introduce them to the liquid agar, and also when we pour the agar into a plate and put the lid on it, we will have minimal condensation. Pour plates are typically used to quantitate or count bacteria in a fluid. Usually you inoculate a known volume of fluid containing bacteria. Typically, this is very precisely measured and dilutions are made very precisely. But we're going to use a less precise method today. We're going to use the drop method. There are about 20 drops in a milliliter. So one drop would be approximately 0.05 milliliters or 1/20th of a milliliter, and two drops would be 1/10 of a milliliter. To start your pour plate, you're going to get a sterile Petri plate and label the bottom of the plate, the part of the plate that's going to have the agar poured into it. Then you're going to turn the plate up so the lid is on top. Then obtain a loop of liquefied agar and add your assigned volume of broth to the culture. You'll take the lid off of the agar tube using your pinky again. Remember, one drop represents 1/20th of a milliliter or 0.05 ml. And two drops represents 1/10 of a milliliter. Gently mix the agar to distribute the bacteria evenly in the agar, and then pour the inoculated agar into the bottom of the Petri dish and replace the lid. If the agar does not quite completely cover the bottom of the plate, very gently swish the plate around in a figure 8 form until the agar completely covers the plate. Allow the agar to solidify and then incubate the inverted plate for 18 to 24 hours at 35 degrees centigrade. Cultures will be examined after they have been incubated for at least 18 to 24 hours. When you examine your broth culture, you're looking for cloudiness or turbidity. That indicates growth. If there is no cloudiness or no turbidity, then no growth occurred. When you examine your agar slant, you're looking for growth on the slant. On this tube, the slant is unchanged, but on this one, you can see growth in the streak line. Broths and slants allow for growth, but they are not isolation procedures. Your streak plate is one of the ways that isolation of bacteria can occur. Because all of the colonies are going to be on the surface of the agar, this allows for viewing colonial morphology. As you look at your streak plate, you are looking for isolated colonies, colonies that are not touching each other. These will have come from a single organism, and so they will be pure cultures. Colonial morphology means that bacteria have different appearances to them. Different bacteria grow different looking colonies on different media. Colonial morphology is a part of bacterial identification. When you give a colonial morphology, you must list the media that the organism was growing on, and you note anything about the colony, like its color if it's on a certain media, or if it's on sheep blood agar, did it provide any kind of hemolysis of the agar. Then you'll want to describe colonial form, elevation, and margin. Colonial form is the overall shape of the colony. If the colony is round and greater than a millimeter in diameter, we say that it is circular. If it is less than a millimeter in diameter and round, we say it is punctiform. And if it's not round, it's an irregularly formed colony. Colonial elevation is the height the colony grows from the surface of the agar. Some colonies will appear to almost be part of the agar. They'll be so flat. We'll call them flat colonies. Most colonies round up a little bit. They are said to be convex. And sometimes you may see a colony that appears to have a point in the middle. That will be umbonate. The colonial margin has to do with the edge of the colony. If the edge is nice and smooth, no matter what the shape of the colony, we say that the edge is entire. If the edge is not smooth but a little bit ruffled, it is an undulate margin. The pour plate is used to quantitate or count bacteria in a fluid. The way this plate was made was all cells were separated from the others in that liquefied agar. Each colony that grows will then represent one cell. So we refer to those as colony-forming units. As you examine your pour plate, you want to try to count the number of colonies. Some of the colonies will be on the surface of the agar. You'll be able to do colonial morphology on those, but others will be embedded in the agar. And those that are embedded in the agar will all have the same shape and size because they were trapped in the agar and couldn't grow freely. Sometimes the use of a Quebec colony counter helps you with counting your organisms. The Quebec colony counter is just something you put your plate on, and you'll have these little squares in the background so that you can count the colonies in a square and then move to the next square and so on. When evaluating the number of bacterial cells in a fluid, we usually use serial dilutions. That is, we take a milliliter of the original solution and we dilute it several times. Usually 1 to 10 dilutions are used. All of the dilutions may be planted, so you'll have many plates. The correct plate to count is the one that has more than 30 colonies but less than 300 colonies. Less than 30 colonies is considered statistically insignificant. More than 300 colonies is too difficult to count. To calculate the number of colony-forming units per milliliter, you take the number of colonies you count on that plate and multiply it times the dilution factor that that plate had. That will give you the colony-forming units per milliliter. This is primarily used on things like water or milk, and largely it's been replaced using spectrophotometry. Let's look at an example of how to calculate colony-forming units. The mixed culture you used in lab, we placed 1 drop in 2 milliliters. 1 drop is 5/100ths of a milliliter, and 5/100ths of a milliliter in 2 milliliters makes a 1 to 40 dilution. Then we took 1 drop of the 1 to 40 dilution, and that was added to the melted agar. Since that 1 drop was 0.05 of a milliliter, that's a 1 to 20 dilution. If you count 52 colonies on your plate after it's incubated, then you want to do the calculation by multiplying the number of colonies times the dilution factors. In this case, if you counted 52 colonies, you had a 1 to 40 dilution factor initially, and then because you only planted 1/20th of a milliliter, you had another 1 to 20 dilution. So 52 times 40 times 20 is 41,600 colony-forming units per milliliter.